So today I'll focus on the work that we've been doing over a number of years on how APOE influences not only amyloid related pathologies, but also tau-mediated neurodegeneration. So here's a list of the disclosures for the talk. And I think, I'm talking to an audience that knows a lot about Alzheimer's disease and related disorders. I think it's important in the beginning just to mention that what I'll be focusing on today is really how APOE influences Alzheimer's specific pathologies. But we know that while Alzheimer's disease contributes probably to around 70% of people that develop dementia, even in those who are diagnosed with Alzheimer's disease and actually have Alzheimer's pathology, there's a number of other disease pathologies that often contribute to the disease, especially in older people. And I'm not going to talk much about that, but I certainly want to point that out. It's not like Alzheimer's disease only clinically only represents contributions from what I'll talk about, but it's certainly an important issue. Okay, so I'm going to sort of set the stage to talk about the time course of pathology, because I think in Alzheimer's disease, because I think it does relate to how one thinks about whether a gene or a protein of whatever type influences the disease. I think it's relevant to think about when it is that it impacts the disease and how does it impact the disease. So we know for a long time that some of the major neuropathologies of this disease are the deposition of the amyloid-β peptide in the extracellular space in plaques, and the accumulation of hyperphosphorylated forms and other post-translational modifications of tau that's aggregated inside nerve cells and their processes. And of course, concomitant, especially with the development of tau pathology, there's a lot of loss of neuron synapses and tissue in the regions of the brain that demonstrate that clinically we see becoming impaired. And we'll talk a lot as we go along about the inflammatory response that occurs when these different pathologies start. This is before the Jack curves became more popularized. We actually showed something, sort of hypothesized something very similar based on clinical pathological, but now biomarker data that I think clearly shows that, at least in regard to amyloid and tau pathology, these occur in bulk in a very different time course in people that develop Alzheimer's disease. Namely, amyloid deposition starts in the neocortex just to build up probably 20, 25 years before symptom onset. I don't show on here the inflammatory component of the pathology, but it also starts around the time that amyloid is developing and changes over the course of disease. But it's only a few years right before clinical manifestations that tau pathology really moves into parts of the neocortex, which corresponds to ultimately what we tend to see clinically in the disease. So if you think about, it used to be, I think it's been hypothesized for a long time now that that amyloid in some way both initiates and causes the disease that we know it. And there's certainly a lot of evidence, genetic and other, that A-beta is critical for this disease, but there's no question that there's many other elements that are required to occur to really develop the disease. And part of that is how amyloid somehow is interacting and influencing tau pathology, and also how inflammation interacts with both of these pathologies. So in relation to this diagram, what I'll talk about the rest of the talk is how APOE appears to influence the process of A-beta aggregation, but also its toxicity. And then the second part of the talk, I'll focus on how it might be influencing an aspect of tau pathogenesis as well. So we know that there are several genes, APP, PS1, PS2, that if you are unfortunate enough to have a mutation in one of these genes of certain types, you will develop Alzheimer's disease at a relatively early age. But of the late onset forms of Alzheimer's disease, which is most of the cases that we see, it's also a very highly genetic disease. And APOE genotype is quite unique amongst diseases that occur, not just Alzheimer's disease, but also other common diseases, in that it's the E4 allele, which is associated with increased risk, is relatively frequent in the population, and it also has a strong effect. So this is the allele frequency, but about 25% of normal people carry at least one APOE allele or two. And you'll see in a moment that it increases risk to AD quite a bit. So we know now since 1993, with many studies supporting this, that APOE genotype affects risk for both late onset Alzheimer's disease, but also cerebral amyloid angiopathy. And it probably contributes to disease in synucleinopathies and tauopathies, although the data for that has not been worked out as well. APOE comes in three flavors, and three common flavors in humans: APOE 2, 3, and 4. And those are due to sequence variation only at two sites, 112 and 158, where the amino acids are simply changed by a cysteine or an arginine between the three forms. And if you have one copy of E4 versus two copies of E3, you have about a 3.7-fold increased risk to develop the disease as a whole across many populations. This is different in some populations than others. And if you have two copies of E4, your increased risk is about 12-fold relative to E33. And E2 decreases risk by about 40% relative to somebody who's E33. So this is a huge, huge risk factor. So the protein itself is 299 amino acids. It's produced at very high levels in the liver and a few other organs, but it's also expressed very highly in the brain, where it's made predominantly by astrocytes, but it's also expressed at quite high levels in reactive microglia. And in the central nervous system, it's only released in HDL-like particles, unlike in the periphery where it's in different size lipoprotein particles. Another interesting fact about it is that if you look at any protein that forms an amyloid, whether it's A-beta or any other protein, it co-aggregates with the amyloid protein that deposits. So APOE clearly has pleiotropic effects in different situations, but I think it's still pretty, one of the most important reasons why I think it's still a risk factor for AD, not necessarily how it affects progression, but a risk factor is because it influences the process of A-beta aggregation and clearance. And I'll just briefly go through some of that and talk more about it as a potential target in regard to A-beta. So if in fact one of the main things APOE does to affect risk is on amyloid, what one might predict if this is a curve of the onset of the different pathologies, that APOE 3 on average people would be like this, and what would happen when E4 is it would drive the red curve to the left so that everything would then happen earlier after that. And E2 would be expected to do this. So what you'd predict then, if you look at normal people hitting middle age, that this is when you might start to expect to see this difference if that's what's going on. So this is just an example of one study that we published. There's several others in the literature where we studied, in this case at our ADRC, over 200 people who were cognitively normal between the ages of 50 and 70, mean age of 60. And we looked at whether or not they had amyloid in the brain, as defined either with CSF criteria or by amyloid imaging. So if you focus on the CSF data, what you can see is that if those that were E44 at a mean age of around 60, already about 65–70% of those people already had amyloid in the brain. And those that had one copy of E4, it's about 30-something%. Or E33 around 20%, and if you had an E2 allele, very few of those people had amyloid deposition. So if you don't ask the question whether APOE is a risk for AD, but whether it's a risk factor for A-beta amyloidosis, one copy increased risk by 12-fold and two copies by 36-fold relative to not being E4. So this, I think, is really strong evidence that this is a major factor as to why APOE is linked with risk. You can model this in animals, and this is just one example. And in one of the APP models that we've used over time, over the years, you can see that if you knock in the human APOE coding sequence into the mouse genome, and you look at mice that are E22, E33, or E44, you see the same kind of effect, that the pathology starts earlier in E4, and at any given time point that's more than E3, then more than E2. And I'm not going to go into the mechanistic studies we've published on regarding why this occurs, but I think it's there's two things probably going on in terms of mechanism. One is that before A-beta deposits, we found that APOE actually is involved in slowing A-beta clearance of the soluble form of A-beta, so that the levels of soluble A-beta in young animals in the interstitial fluid of the brain are higher with E4, somewhat lower with E3, and somewhat lower with E2. And it's probably by APOE and A-beta competing for similar receptors. But the other thing that probably underlies this mechanism is that APOE can ultimately bind to A-beta that becomes aggregated, and it probably plays an important role in seeding. And there's a number of in vitro papers that have seen this, and I'll talk about this in the context of the in vivo situation in a moment. So in regard to this phenotype of A-beta aggregation, if you wanted to target APOE in some sort of therapeutic way, would you want to increase or decrease the level of APOE? So in this genetic experiment we did, and also, which was also done in a similar paper at the same time by the Gladstone Institute, we basically took APP PS1 mice, and we simply asked if they have 2 copies of E3 or 2 copies of E4 versus 1, what happened to amyloid deposition. You can see that clearly, if the animal throughout the whole life had 50% less APOE, there's a lot less development of A-beta deposition. And now this is throughout the life of the animal. So one of the things that we asked was, well, okay, what if you lowered APOE during life, would it have a similar effect? So we, Fat Huen, an MSTP student in my group, worked with Ionis Pharmaceuticals and developed an ASO to knock down APOE. And in this set of experiments, he showed that if you inject the ASO intracerebroventricularly at either P0 or out at 12 weeks, you can lower APOE levels by about 50–60%. So he then, we designed this experiment where we took these APP mice that had E3 or E4, and we injected the ASO either right after birth, or in other experiments I'll show you in a moment, we injected them later after A-beta deposition just began to form. So if you inject and you lower APOE by about 50–60% right after birth, you lower amyloid deposition by about half. That's kind of similar to the genetic experiment. The other thing to point out, though, is that if you look at the dystrophic neurites that are around plaques, you can see that there's a lot of them, as stained here with LAMP1, which is present in the dystrophic neurites. But it also, on a per plaque basis, lowering APOE 4 or 3, lowered the amount of damaged axons and dendrites surrounding plaques by a lot, about 50%. And so that suggests maybe there's another effect of APOE, not just on the amyloid itself, but somehow on the toxicity, the local toxicity that amyloid is causing. So in the other experiment that was done is we lowered APOE later. So this now is until just after 6 weeks of age, when A-beta deposition in this model has already started. And when we do this and then look at 4 months of age, we actually see no difference in the amount of amyloid deposition in the brain. And this, along with another set of work that Guo Junbu's lab published concomitantly, we suggest that what's going on is that APOE is very important in the initial seeding of A-beta, but once A-beta actually is deposited, it doesn't have as much effect on the growth. At least by lowering it 50% or so, it doesn't affect the further growth of plaques much. However, having said that, we still saw this very big effect on lowering the amount of dystrophic neurites around plaques. So again, this suggests a potential additional role for APOE in somehow leading to the damage to neurons. And this, I think, will come back towards the last part of my talk, where I look at the effect of APOE on neural damage in the setting of tau pathology. So one of the things that is interesting about the cells in the brain that do make APOE is that it turns out there are probably some differences in the APOE that's released from the cells. So in this set of experiments, Tom Mahan, a PhD student, was looking at APOE that was released from either cultured astrocytes from these APOE knock-in mice or cultured microglia. And what you can see on this non-Denitrian gradient gel, which looks at the size of APOE lipoprotein particles that are secreted by these cells, is that the astrocytes, which has been known for some time, secrete APOE in a relatively normal size HDL particle that's about 12 to 20 nanometers in size. But the microglia actually secrete APOE at least in vitro, in a less lipidated or smaller particle. So that means that the amount of lipid, and probably we're looking at this now, the types of lipid that are in the particles is going to be different. And that might be very important in regard to the function of those particles. So I bring that up because one of the things that we've recently done is to look at the effect of knocking out only astrocyte APOE in this model, in these mice that develop amyloid deposition, to ask, what does that do to these different pathologies that I was just showing you. So in these experiments, Tom Mahan made APP PS1 mice that have E3 or E4, but their E3 or E4 alleles are flocked, so that if we add a Cree, then it will remove APOE from whatever cell type that we're turning on the Cree in. So in this case, we use mice in which the ALDH1 Cree ERT transgene is present, so that if we give tamoxifen, we can remove APOE from astrocytes. So in these experiments, tamoxifen was given to the animals at 4 weeks of age that either had the ALDH1 Cree transgene or not. And then we looked at 4 months of age. And so what you can see is that when we knock out astrocyte APOE, we lower APOE levels about 60–70% or so. At the mRNA and the protein level. And if you look at amyloid deposition with this X34 dye, which stains A-beta fibrils, you can see there's a huge reduction in amyloid deposition when we remove astrocyte APOE3 or astrocyte APOE4. And in fact, the phenotype, at least at the level of amyloid deposition, is very similar to a complete APOE knockout. So I think that argues, at least for this particular pathology, the astrocyte APOE really is a key factor. And I know that in a recent paper that came out of UC Irvine, where at least in APP with mouse APOE, that removing microglial APOE did not have a big effect on this kind of pathology. So this is from Kim Green's work. So, okay, so having said all of this, it looks like probably through A-beta affecting APOE affecting the seeding of A-beta, but also by affecting its clearance through receptors, that influences A-beta aggregation, and possibly somehow downstream there are some other effects that APOE has on toxicity. So one of the other things that's been known for a long time is that, from early 90s, is that APOE co-aggregates in amyloid plaques and in cerebral amyloid angiopathy. So a number of years ago, we started to wonder whether if we had an antibody that actually bound to APOE that was selective for the APOE in plaques, that might actually be another way to get rid of this pathology. Perhaps it would have a somewhat different effect than just using an antibody, for example, to the A-beta peptide. So we collaborated a few years ago with Denali Therapeutics, and we had created some antibodies to APOE that appeared to have some interesting properties. And so I'll just go through this to then get to why this might be relevant even therapeutically in the future. So we wanted to assess whether antibodies against human APOE could reduce amyloid pathology and to understand the mechanisms how this might work. So in some initial experiments we did, we took these APP mice that, in this case, they express human APOE4, and we gave them different antibodies to A-beta or to APOE. And one of the antibodies shown here reduced A-beta pathology to a somewhat similar extent as some of the A-beta antibodies that have been published. So we started to characterize this particular antibody and some other ones to a greater extent. And one of the interesting things is of the different antibodies we made, this is just two here, is that these antibodies bind to APOE that's put on an ELISA plate. Were common in APOE quite well. This particular one I'm showing you, the HAE4, it binds to E3 and E4. It actually doesn't bind to APOE2 because of its binding site encompasses around position 158, which is different between E2 and the other isoforms. But one of the interesting things we found is most of the antibodies to APOE that we had produced bind also very well to lipidated APOE, which is the physiologic forms of APOE that are made present in the body. But this one that we showed reduced amyloid, as I just showed you, actually does not bind to lipidated forms of APOE at all that we can detect. So suggests it binds to a particular conformation that's present when this molecule is not folded in its physiological way. What we found, but what's interesting is that this antibody does bind to APOE that's in amyloid plaques, as I'll show in a moment. So we wanted to know, is the mechanism by which this antibody works, does it require microglia, but the antibody binding to microglia stimulating phagocytosis? So we express this antibody in the brain using an AAV construct with either the regular full-length antibody, which in this case is an IgG2AB subtype, or we mutated the FC domain at a single site so that it wouldn't bind the FC gamma receptor on microglia. And when you do this, you can see that this antibody only removes amyloid if it does contain an intact FC gamma receptor binding domain. So implying it really does require microglial mediated phagocytosis to have this effect. So this brings us to the next question we wanted to address, which is an issue that has to do with APOE and cerebral amyloid angiopathy. So we know that APOE is a risk factor for CAA and for cerebrovascular dysfunction in humans. And a number of years ago, we actually looked in one of the first produced APP transgenic mice, the TG2576 or APP Swedish mouse. And this particular animal, unlike some of the other APP mice, develops a fair amount of CAA, especially after a year of age. And what we found back then is that if these mice like humans, some of these vessels with CAA develop hemorrhages around them, microhemorrhages. And you can see this in humans that have AD and also CAA. And if we removed APOE from these mice globally by knocking it out, there's no CAA ever develops. So APOE is absolutely essential for the development of cerebral amyloid angiopathy. So, so why is this important? Well, as you are all probably aware, one of the things that occurs with the advent of the development of anti-amyloid antibodies, which have been put into humans now for the last number of years, is that many of the antibodies that are capable of removing amyloid from the brain also are associated with this side effect called ARIA, amyloid-related imaging abnormalities with edema or hemorrhage. And these are just some of many of these antibodies, not all of them, have been shown to have this particular side effect in a subset of people. And this side effect is for sure due to the fact that in some way the antibodies are interacting with amyloid angiopathy and inducing this problem. So what's the cause? Well, we don't know for sure, but we know that A-beta antibodies that some of them can solubilize plaques, and that maybe then they get this A-beta gets trafficked through the perivascular space that might disrupt the vascular cells. Another possibility is that the A-beta antibodies with effector function might be recruiting microglia that could cause focal inflammation on target engagement. And there may be other possibilities. So we wanted to know whether this APOE antibody that I just showed you some properties of would have a similar mechanism of action in regard to the effects on CAA specifically. First, we wanted to have a comparator. So we actually cloned the murine human chimeric version of aducanumab so that we could compare the effects of that antibody, which we know can remove plaques, but it also can induce ARIA. Just to show you that if we give this antibody to 5XFAD mice that develop brain amyloid, that as Biogen has shown, this antibody can decrease A-beta deposition by quite a bit. But in order to test the effects of these different antibodies in CAA, we had to create a different model or use a different model. So one of the things we did is we took one of the 5XFAD transgenic lines that Bob Vassar had created a number of years ago that expresses a little bit lower levels of the transgene and starts to get amyloid deposition around 5-6 months of age. And we crossed this animal to E4 so that it expressed, instead of murine APOE, expressed human APOE4. And when you do this, it actually drives the pathology from more in the parenchyma to mostly in the vasculature. So you can see on the bottom that about 85% or so of the A-beta, instead of being in plaques, is now along arterioles, which is where A-beta forms when it forms CAA. So we began to use this model to study the effects of these antibodies. And so we waited till these animals were 8 months of age when they have already a substantial amount of both CAA and some amyloid deposition in the brain. And we gave them these different antibodies, either the APOE antibody, HAE4, or this aducanumab chimeric antibody. And what you can see is that there was a significant decrease, not just in parenchymal A-beta, but also in CAA in the animals that were given the HAE4, the APOE antibody, but there was no significant reduction of CAA in the mice given aducanumab equivalent. And actually, Biogen had previously published that in mice that develop amyloid deposition, you also don't remove the CAA from the model that they had used either. So this seemed to be some differences between the effects of the antibodies. And this is just showing the thioflavin staining of the same animals. So if you look at the properties of these antibodies, you can see that both in mice as well as in humans tissue, we can see that this APOE antibody stains the core of A-beta plaques in CAA, but an antibody to A-beta like aducanumab will stain all the A-beta that's deposited. So it's staining probably because APOE is only a small fraction of what's deposited in these pathologies. So we were interested to look at, what about this potential mouse version of ARIA? So it's difficult to see the edema part without probably doing MRIs, but what we can determine is that some of these CAA pathologies also develop microhemorrhages, and you can detect this with a Prussian blue stain, which stains the hemosiderin that's around a vessel where there's been some small rupture. And so you can see that in the animals treated with control or the APOE antibody, you can still see microhemorrhages, but the APOE antibody does not increase the microhemorrhages over baseline. Whereas the aducanumab antibody more than doubles the amount of microhemorrhages. And you also see larger microhemorrhages in the presence of the aducanumab related antibody. So, so that suggests, the side effect might, if this was moved in, for example, something like this was moved into people, maybe you wouldn't see that kind of a side effect. The other thing also is that we wanted to look at vessel function. So Monica Xiang, who carried out most of these experiments, a graduate student at the time, worked with Greg Zipfel's lab to look at the ability to look at these vessels in living animals through the skull, where you would treat the animals with antibody or not, wait 2 months, and then under anesthesia, thin the skull, and then be able to look at the vessels after you give different chemicals or CO2 that would cause vasodilation of arterioles. And we know that what CAA does when it forms is it prevents the normal vasodilation of arterioles. So in looking on the surface of the brain, just under the surface, right on the surface of the brain, you can see that many of the vessels have CAA. And under basal conditions, you can see the width of the arteriole. And then if you do things that cause vasodilation, like apply acetylcholine, you can see that results in dilation of the vessel over baseline. And this occurred to a reasonable extent in the animals that were treated with the APOE antibody, shown here. But the aducanumab did not significantly increase the ability of the vessels to vasodilate. And this wasn't quite significant with another vasodilator, SNAP, but with hypercapnia, you could also see this effect, especially in the smaller vessels, of an improvement of the ability to vasodilate. So finally, one of the things that we're interested in is, well, why would you get this potential beneficial effect with this one antibody, which we think is also both removing A-beta around the vessels better than another, but why would you get the side effect with one but not the other? And so we thought, well, maybe the inflammatory response has something to do with this. And so we gave these antibodies to these animals and just waited a very short time, like 12 days, and then looked at the expression of different microglial genes, astrocyte genes, and other genes. And you can see that a short time after giving the APOE antibody or the A-beta antibody, there was an increase in microglial reactive type genes, astrocyte reactive type genes. So no major difference between the groups. So that didn't seem to explain perhaps this side effect issue. But one of the things we did notice, if we did the same experiment and waited 8 weeks and then looked at these types of genes, that there was still a persistent microglial reactive signature and astrocyte reactive signature in the animals treated with aducanumab, and that mostly had gone away in the mice treated with the anti-APOE antibody. And so we wonder, perhaps this is happening because there's so much of this A-beta antibody, the aducanumab, that's probably stuck to the A-beta, and there's so much of it there that maybe that's somehow leading to this persistent response. Whereas with the APOE antibody, it's such a small component of plaques, maybe it allows the microglia to do what they need to do, but then not stick around. That's one possibility. And if you actually look anatomically at the blood vessels of these mice that are treated with these antibodies, after 2 months of treatment, there's still a very strong increase of reactive astrocytes in appearance around the arterioles, and this is mostly not present after we use the APOE antibody. So we don't know the detailed molecular reason for this, but I think it really does suggest a persistent inflammation that these A-beta antibodies are leading to around vessels that might be responsible for this side effect. Okay, so I'm gonna change gears for the last part of the talk and really focus on the second I'll just call it the second phase of AD pathology, more when tau is moving into the neocortex. So one of the issues is it possible that APOE influences not just when amyloid starts to develop, but also, let's call it the tau phase of disease, when really neurodegeneration is kicking in, you're starting to lose neurons, losing synapses, and getting symptomatic. And if so, how does that occur? So in order to begin to study this, we thought, well, we need at least to use a model, some sort of model that develops tau pathology, but also develops neurodegeneration. And there's a number of these animals that have been created over the years. One of the first ones made was by Virginia Lee's lab, where they used one of the mutations in tau that was shown to cause a rare form of frontotemporal dementia, where they overexpressed this in the brain in PS19 mice. And And what they started finding is that these mice start getting tau pathology with aggregated hyperphosphorylated tau around 5-6 months of age. And then as this pathology progressed through in certain parts of the hippocampus and entorhinal piriform cortex, you'd start to get loss of brain tissue, which was concomitant with losing neurons and synapses in those regions. So we thought this model might be a really good one to assess not just the effects of something on tau pathology, but also the damage that is being somehow related to the development of the tau pathology. So Young Shi, then a graduate student, led these experiments where basically we just crossed these tau T mice to mice that express human APOE2, E3, or E4, or no APOE at all. And so what was surprising when we took the animals' brains out at about 9.5 months of age, when these mice have a lot of neurodegeneration, for example, in the hippocampus, entorhinal piriform cortex, amygdala, you can see that there's a major damage that's occurring in those brain regions with concomitant ventricular dilatation, but that with E3 and E2, the damage is somewhat less. And what's really striking was that there was almost no damage at all in the absence of APOE. So we said, well, that's interesting. What's going on here? So one of the first things we did is we looked at the inflammatory response. And in this particular experiment, which was done in collaboration with Oleg Butovsky's lab, he looked at gene expression in the brains of these animals at 9.5 months of age. And you can see in the tau E4 mice, each of these lanes is an individual animal. There's a big increase in gene expression in this, what we then called cluster 1 type genes, and a big decrease in these cluster 2 type genes. And this pattern of microglial related gene expression was mostly blocked in the absence of APOE. And we didn't see that pattern at all if the tau transgene was not present. So what's going on with these genes? So it turns out that many of the top cluster 1 genes are subsequently what many people began to call disease-associated microglial genes, such as complement C3, TREM2. And these cluster 2 genes are now what many people call more homeostatic related genes in microglia, things that regulate lipid metabolism like LDLR, SREBP2. And if you stain the brain with a marker for activated microglia like CD68, you can see this huge increase in the tau E4 mice, which is mostly blocked in the absence of APOE. So what about the astrocytes? We see something similar in that, and these experiments were done at the time with Shane Littleow when he was still in Ben Barris's lab, where they looked at gene expression for reactive astrocyte type genes. And you can see that in the tau E4 mice, there was a big increase in these types of genes that was, again, mostly blocked in the absence of APOE. And this is a GFAP stain showing this very strong effect in the tau E4 mice. And in the absence of APOE, there's very little of this reactive type astrocytosis. So it's certainly possible that the reason somehow APOE is modifying the innate immune response in relation to the development of tau pathology. But one of the things that we wanted to ask was, well, do you see this? Can you see this kind of a phenotype when you just have isolated brain cells and not in the context of the in vivo situation? So we cultured P3ONS expressing neurons, which are here labeled in green, on a bed of mixed astrocytes and microglia. And what you can see is that over a few week period of time, if the glia did not make APOE, the cells looked healthy and had good looking axons and dendrites. But if the glia expressed APOE4, over time, they began to degenerate. And this was less so with E3 and E2. So I think this doesn't tell us the molecular mechanism, but it says that probably glial secreted or somehow glial produced APOE can lead to a similar kind of phenotype ex vivo. We also, in response to one of the reviewers for this paper's questions, they asked, well, because the model we were using is of course, it's a model of primary tauopathy, it's not an AD tau mutation, is there any evidence that APOE genotype is linked with something about disease risk or progression in primary tauopathies? And we certainly weren't able to answer this completely, but one of the things we did is collaborated with Bill Seeley at UCSF and simply asked the question from a large brain bank he had of people that died of primary tauopathies like CBD, PIX, and PSP. If you normalize different brain regions for the amount of tau pathology, was there a difference in the amount of local brain injury in the presence of different APOE alleles? And that's pretty much exactly what he found. So there was generally more damage when you normalize for the amount of tau pathology in these primary tauopathies in the presence of E4. So we also did a set of experiments where we looked at people in our ADRC who were very mildly impaired, and all of them had amyloid deposition, and then followed their course over time and noted that the progression of disease was significantly higher on an APOE dose in an APOE4 dosage dependent fashion. Not dramatic, but it was definitely significantly faster. So I think this argues that that one of the effects of APOE might not just be on amyloid, but also on this sort of damage linked to tau pathology. So in terms of, translationally, is this something that you could actually approach and do something about and have an effect? So we basically took these same tau E4 mice, used the same ASO to APOE that we'd used before, and waited till these mice were 6 months old when they already are developing some tau pathology and also a little bit of neurodegeneration, and then gave them this ASO starting at this age, and then followed them out to over 9 months of age. And as we showed before, that lowering APOE will lower, or giving the ASO will lower APOE levels. And when we look at neurodegeneration as marked by brain atrophy, you can see that in the animals given the ASO, there was a significant increase in both hippocampal and piriform entorhinal cortex volume, and the ventricle did not dilate as much, presumably because there's less atrophy. So I think this is something that might be this kind of thing, whether you lower APOE in this way or with gene therapy or other mechanisms. I mean, it does suggest this might be something that could be done a little bit later when the disease is just kicking in clinically, and maybe this might be worth assessing in some way in trials. Other evidence that lowering APOE could be protective is shown here. And again, these are experiments done by Young Shi, where we basically took the P3NS mice, in this case expressing mouse APOE, and we simply crossed them to mice that overexpress the LDL receptor in the brain. And we know that these particular mice have about 90% lowering of extracellular APOE. And when we did that, there was, again, a very strong protective effect against tau-mediated neurodegeneration. So now whether this occurs with APOE2, we don't know yet, but certainly with APOE3 and 4, we have other experiments we haven't published where lowering both of those is neuroprotective in models of tauopathy. So I mentioned earlier that, of course, APOE is expressed at high levels in astrocytes, and but it also is expressed in reactive microglia. And this shows some data from wild-type mice, and this is a logarithmic scale where we did single nuclear RNA sequencing from hippocampus of wild-type mice, and each of these columns represents a different cell type. It's mostly neurons, but this column is astrocytes, and this is microglia. You can see under basal conditions on this logarithmic scale, APOE is made by astrocytes more than 100-fold higher than other cell types. Actually, 15 is oligodendrocytes. But where there's damage in the brain, in this case due to tau pathology, you can see that the reactive microglia start making about as much APOE on a per cell basis as astrocytes. So like I showed you earlier, one of the issues is, okay, so APOE is having these interesting effects, not only on A-beta, but also on tau-mediated neurodegeneration. So if APOE, whether it's made mostly by astrocytes or in some cases some by microglia, does that effect of APOE require microglia in order to see neurodegeneration? So to address this in a very blunt way, we used one of the CSF1 receptor inhibitors called PLX3397, and Young showed that if you give this orally to mice, you can remove most of the microglia once we use a high enough dose. And if you take these TAUE4 mice that I just showed you, as we've shown previously, there's a lot of damage in the brain that occurs. And if she began to deplete microglia from these mice at 6 months of age and then followed them out to 9 months of age, that completely blocked all the neurodegeneration that we see in this model. This is a massive protective effect. And very similar results were recorded by Mancuso et al. In a different P1L model a few years ago as well. And then, of course, as we had seen previously, if you remove APOE, you get a very similar effect. If you remove APOE and remove microglia, you get a similar effect. So, you know, this is a very blunt experiment, but I think it shows at least in the setting of tau-mediated damage, that microglia are obviously important in the injury. So that leads us to one of the final things I'll talk about. So what about this whole APOE being made by different cell types? Which of those plays a role in the damage that we see due to tau? So these are experiments led by Chao Wang in the lab, and we basically made these tau E3 and E4 mice that I showed you earlier, in which we can remove APOE by expressing Cre. So we bred these mice to ALDH1 Cre ERT mice, so that if we gave tamoxifen, we could knock out APOE from astrocytes, like I showed you earlier in the amyloid mice. So we gave the tamoxifen or oil starting at 5.5 months of age, when tau pathology is just starting, and then assess the animals at 9 months of age. So as I showed you earlier, we can decrease APOE levels by about 60% or so when we give tamoxifen. And while APOE is shown in this single nuclear RNA sequencing experiment to be mostly gone, it's actually increased in microglia because we're not removing the microglial APOE. When we look at one of the main phenotypes, which is just brain atrophy, you can see that in the oil-treated TAUE4 mice, there's a lot of damage. When we remove astrocyte APOE4, there's significant neuroprotection of the regions of the brain that degenerate. The APOE3 mice have some injury, not nearly as much as in the E4 mice. And when we remove APOE3, there's not more damage, but there really wasn't a significant effect on neuroprotection. It could be that there's just not that much damage, so there's not as much to protect, or maybe something else is going on that's unique with APOE4. We also see a functional effect when we remove astrocyte E4 in this particular nesting behavior. 4 to 5 is a normal score, and you can see these animals are quite impaired, and when you remove astrocyte E4, it gets them much closer to normal in this particular behavior. One of the things that's interesting is if you remove astrocyte APOE3 or 4, there's not a big effect on the amount of tau that accumulates in the brain. There is somewhat of a decreased level of AT8 staining, but you can see biochemically there's not a really big effect on tau, implying that the protective effects of removing APOE4 are more likely through probably affecting the immune response. We did a lot of single nuclear RNA sequencing from these animals, and I'm not going to go through all the data that we've published. You can read about that, but I think I'll just show you some the data from the microglia. So there are sort of 3 populations of microglia, but number one here are the more homeostatic type microglia, and what's labeled as 0 are more the disease-associated microglia. What you can see is that in all the tau mice, this black bar represents the disease-associated microglia. And by removing APOE3 or 4 from astrocytes, you really don't see any major effect on the total proportion of cells that obtain this phenotype. However, but when you look at individual gene expression of these disease-associated microglial genes, many of them are decreased in the total level that they express when we lower APOE4. And this is really illustrated well when you look at sort of a classic homeostatic microglial protein like P2RY12, where in the tau E4 mice, this expression is low. When you remove astrocyte E4, it increases quite a bit. And you see a yin-yang effect in that the disease-associated microglial marker CLEC7A is high in the tau E4 mice, and then it's strongly lowered when you lower astrocyte APOE4. And then finally, if you look at synapses in these mice, you can see that staining with a postsynaptic marker PSD95, there's a decrease in the synaptic puncta in tau E4 mice that is improved when we remove astrocyte E4. And then if you look at the microglia in these regions, you can see that there is phagocytosis of PSD95 positive synapses by microglia. And if we lower astrocyte E4, you actually see a decrease in synaptic phagocytosis that's illustrated in this graph. So I'll end the talk just by summarizing the last part. I think it looks like APOE, especially E4, really drives tau-mediated neurodegeneration, at least in this model system, and that microglia are required for this process. But that APOE4 produced by astrocytes really appears to play an important role in this neurodegeneration. We're in the process of trying to figure out if microglial produced APOE has any effect in this model system. We don't know the answer yet. So in addition to assessing the role of APOE in microglia, I think understanding how astrocyte produced APOE via interactions with other molecules and cells, such as microglia, contributes to neurodegeneration is going to be important to sort out. So while we don't understand the mechanism, it could be that APOE that's secreted by astrocytes obtains a sort of a toxic property, as recently suggested by some experiments published by Shan Littleau, or alternatively, perhaps APOE in the setting of damage to synapses might serve as an opsonin, maybe like C1q, and there are receptors on cells that then modulate some of that synaptic damage. So these are things I think that need to be sorted out in the future. And so I will stop there. I think I tried to thank many of the people along the way, but we certainly have time to take some questions, hopefully. So thanks.
Thank you for a wonderful talk. A lot of really important insights for us. I think we're open for some questions. There's a couple in the chat for you too that you can talk about if others don't speak up.
Okay. Do you want me to go through those or have people just unmute?
Let's have people unmute first and then we can get to them maybe if. Okay. Yeah, I think one of the questions in the chat, Gregory Brewer has a question for you. So why don't you jump in, Gregory?
That was a real tour de force for a lot of control of APOE expression. Very nice. With the redox active cysteines that are specific for APOE4, what changes have you seen or do you speculate would changes in the redox status of the brain have on APOE function?
Well, that's a great question. I think that's still one of the possible reasons for some of the isoform differences that's still never been completely clarified. One of the ways we're trying to look at that now is there's a lot of recent data suggesting that how APOE influences lipid metabolism in the brain is by affecting glial lipid metabolism in particular. And so we've been doing some experiments to look at the lipidomics of glia in these model systems that I just showed you. And one of the things that we're starting to see is that in the presence of the different APOE isoforms, you tend to see patterns of lipid changes, especially in microglia when damage starts to occur, that suggests that some of them might be due to changes in reactive oxygen species that are occurring differently based on the isoform. So I don't have a final answer to that, but I think it's still possible that those differences in the cysteines could account for maybe the differences in glial, not just the lipid metabolism per se, but the lipids that might be important in reactive oxygen species related changes.
Dr. Manucchi, you had a question?
Yeah, thanks, Bryce. David, thank you, as always, for the great talk and very clear data and messages. I was curious on something you more alluded to, which was, it sounded like some of your thoughts and considerations about the APOE antibodies as potential therapeutics. And I'm sure you've probably thought through maybe the different pros and cons of that approach. I wonder if you could kind of share some of those thoughts with us.
Right. So I think, well, first, I think removing amyloid from the brain, however one does it, certainly still is something that I think could have a big impact on AD, especially if you just started earlier than most of the current trials were started. So I think because the APOE antibody can also do that, I think that might, so why would it be different than just using an anti-A-beta antibody? And I think the side effect issues, one, we don't know if that will occur. The side effect, lack of side effects so far in the model we use would also be present in humans or not. That would have to be tested. But I think from some of the things people have asked a lot about this, well, why wouldn't, if you gave an antibody to APOE, why wouldn't it influence, all the APOE that's in the body in some way? And I think the reason it doesn't appear to do that is that this antibody simply doesn't bind the forms of APOE that are present in the body, because they're all lipidated, and whatever the conformation of APOE that's in the presence of lipid is different. It's completely different than when it misfolds, and it appears to misfold in plaques. Now, maybe we need to certainly look for more evidence that it really doesn't affect normal APOE, but so far we haven't been able to find that it affects APOE, for example, in other organs or even the brain for that matter, other than what's present in co-aggregated with amyloid.
So is it preclinical work with this kind of moving forward, and is it advancing at all at this point?
Yeah, a company licensed the antibody recently called NextCure, and they're advancing. They're going to determine whether to advance it now into human studies.
Okay, glad to hear. Okay, thanks.
That's very exciting. There's one more question that's in the chat. For you, the first question that was put in the chat. And I think probably just for sake of time, maybe we can answer that and then probably call the time.
A question from Michael or a different one?
Yeah, from Michael.
Yeah.
Well, I guess we have another one from Grant as well. We'll do Michael's.
Okay. Yeah, so it says how did we generate this antibody, and did we map the determinant on APOE4? Yeah, so we know that this antibody, we just made a like standard mouse monoclonal antibody where we injected recombinant APOE that was not lipidated into APOE knockout mice, and then generated monoclonals and then screened them. So most of the antibodies are not like this antibody, most of them do recognize normal forms of APOE like secreted by cells. This particular one and one or two others did not, but they did recognize, they certainly bound APOE, but only when it was non-lipidated. This particular epitope of this antibody is surrounding the 158 position, which is why it doesn't bind. So it must bind in the arginine plus other amino acids in that location, but there's a cysteine in E2 there, so that's probably why it doesn't bind E2.
So David, they are not binding to lipidated.
Correct.
APOE4. But then how you can expect that they will work when you are injecting in vivo?
Well, how do they work? Well, I think they bind to the APOE that's in the plaque, and then just like with A-beta antibodies, many of them, they will allow microglia to phagocytose the material. I think that's.
Did you check the binding to receptor? Could you block the binding to receptor?
You mean FC gamma receptor, or?
Yeah.
Well, we didn't. Yeah. So the experiment we did is we mutated the region on the antibody that allows binding to FC gamma receptor, and when you do that, you can no longer see an effect.
Thanks, David.
Yeah. All right. So last question, Grant McGregor.
Thanks, Bryce. That was great. I really enjoyed that. Thank you. It struck me that in many of your studies, you're comparing the humanized APOE3 or APOE4 with against a null. And the sort of tantalizing thing that is admitted that I'm interested in is the wild type mouse. And so from a modeling perspective, seeing as you're a guru of APOE, I wonder if you can comment on what your feelings are regarding the optimum APOE to have in a mouse model of AD. Should it be should you leave the mouse APOE, which is, maybe something completely different from the human APOE, or is it better to use a knock-in of APOE4?
I think it really depends what you're asking. So mouse APOE is highly homologous, but there are a number of amino acid differences. So many of the functions are very similar to the human version. So for example, it depends a little bit on the phenotype. So mouse APOE, if you take these amyloid mice with just have mouse APOE, obviously they get pathology and if human APOE, actually mouse APOE is actually more amyloidogenic to A-beta than the human forms. So in terms of just having that phenotype, I think it's certainly reasonable to study the animals with mouse APOE, but it depends what the question is you're asking. If you're actually trying to understand what human APOE is doing as well, I mean, that could be certainly an advantage of using the humanized mice. Subsequent, also in the tau model that we're using, if there's mouse APOE there, they certainly get a lot of tau pathology and neurodegeneration, like I showed you with same as with human APOE. But in that case, APOE4 tends to even cause a somewhat greater phenotype at any given age, whereas E3 and E2 are more similar to the mouse APOE in terms of that particular phenotype. So I think it really just depends on what the question you're asking as to whether, whether you want to study the human version or the mouse version. But if you're ultimately going to do something that might be relevant in humans, I think it's certainly reasonable to study the humanized mice because there are some subtle differences in the function.
Do you think differences in the way that the mouse and human proteins fold because of some of the amino acid changes could impact the mechanism of spread of tau?
That's a good question. I don't know, but one of the things that in this field, just as background, the crystal structure of the N-terminus of APOE was shown 30 years ago. But even though that structure is correct of what they crystallized, a very nice paper, the structure of APOE in vivo is not that. It's going to be completely different because when APOE is on a lipoprotein particle, it's completely folded differently than when it's not with lipid. So one of the things we've been working on really hard the last few years, and this is not a trivial problem, is to really understand what is the molecular structure of APOE when it's on a lipoprotein particle. So we've been using cryo-EM to try to understand that, and we're getting closer, but I think ultimately, if you really know the positions of all the amino acids and the structure, then we really can sort of try to map out try to answer the question that you're asking.
Thank you.
Thank you, and thank you for everyone for being here. Thank you, David Holtzman, for giving us a lovely talk as always. It's wonderful to see your work. One last thing for everyone, just